But yeah, thank you all for coming today. I reached out to some folks in the department to see if anybody wanted to give a talk, but I think there's still folks coming back from break and whatnot. So I'm just going to this month talk about some of my stuff, and then once the semester is fully started, I think we can probably get some other folks to join in. But yeah, so today it's not an incredibly long talk, but I really just want to talk a bit about um, this work I did during my master's, which I think is relevant right now, even though I will admit it's in Kentucky, but the animals that I dealt with, you can find all of them in Ohio around here. So I feel like if we just don't think about Kentucky, if you replace that with Ohio in your brain, then it's still it's still ecophilia relevant. But yeah, I'm going to talk a bit about this and see what y'all think about it, because this is actually, I'm in the process of publishing this right now, and I think it's good to get some feedback on it. So let me see. There we go. Okay. So, as far as my sort of focus with my work is in a word is predation. And as far as amphibians are concerned, we can divide up predatory effects into consumptive and non-consumptive effects. In the sense that, consumptive effects, I just have a little frog there eating a worm. But, by eating, consuming things, we see, they're reducing the population size, the abundance of prey. You reduce species richness potentially in that way. And you can alter ecological functioning via, if you are a top predator, you're affecting the whatever you're eating below you, and that whatever that eats is being effective, and so on and so forth. But what a lot of folks don't or aren't familiar with are non-consumptive or indirect effects. In the sense that, just by being present, a predator being present, not eating anything, if we just focus on, say, a frog and an insect of some kind, then if a frog's just present or a predator's just present, that can limit the growth of prey, because the prey aren't going out and eating as much. They're staying in their little holes, their little shelters, and they're scared. They're not doing as much. So that is a little bit about both those types of effects. So it's not just that. If we just think about it in a vacuum, it doesn't tell us all that much. Any predatory effect is going to be influenced by a variety of biotic factors, such as, for example, larger predators are going to probably be eating more. Predators, this is a really understudied topic, but predators that have a maybe more aggressive temperament are going to be eating more or causing more damage, we can say. But there's also environmental factors. So we can think about abiotic factors, in the sense that, for one, especially among cold-blooded animals, if it's warmer out, we all know climate change is happening, especially up, in northern areas like ours. If temperatures are warmer, we're going to probably see that these cold-blooded animals are, predating more, they're eating more, they're having a greater consumptive effect. But also things like habitat shading. If there's less light coming down into a given habitat, say it's an aquatic habitat or wherever, there's less light. So you're going to see, maybe predators can't, if they're very, if they rely on their eyes a lot, they can't see prey as well. Maybe they're not eating as many prey. So what about in tadpoles? Because that's sort of my bread and butter is amphibians, and I thought for my first project here when I was doing my master's, when this paper has since been published, what about tadpoles? They're really classically portrayed as being these sort of just like scum eaters, sort of pond scum feeders, most of them. But there's a lot of cases in which I showed where frogs around here, for example, your bullfrogs, your green frogs, your wood frogs, they're tadpoles, not even the frogs yet, but the tadpoles are eating a lot of animal matter, whether it's zooplankton or larger macroinvertebrates like worms, like aquatic worms. They're really, especially in forested environments, they really could be the dominant vertebrate consumers in these freshwater systems. And studies in other freshwater systems, getting back to, what other factors are influencing this. Other systems for other groups indicate that predator body size and degree of habitat shading, how much light is coming into the habitat, are really important to influencing predation rates and detection of predator cues by prey. So, why, sort of ending my sort of little background segment here, why even study these effects in tadpoles specifically? Like, what's so important about them? Really, it's more, what is affecting amphibians. So, for one, climate change is driving all kinds of changes in, not just temperature, but through that, animal body size, specifically amphibian body size, and canopy coverage. For example, a lot of ectotherms are having smaller bodies as climates warm, faster growth rates, but in many cases, smaller bodies. So how's that affecting predation rates? And canopy coverage, if it's drier under warmer temperatures, if we have less rain, it's hotter, maybe we don't have as much tree growth. Maybe there's less canopy coverage, more light coming into these aquatic or any kind of so, and I bring that up just because, amphibians in particular, which I'm investigating here, is my sort of focal group, nearly 40% of amphibians, it may have reached 40 by the time when I, 3 years ago when I had this data, it may have very well reached 40% by this point, but they're at great risk of extinction. And they play a variety of important roles. Just one, for example, I mean, they're one of the few vertebrate groups that you see that pretty much throughout the whole group having an aquatic stage and a terrestrial stage. Not for every one of them, but for most. And they're really, they're cycling those nutrients between aquatic and terrestrial habitats. You may have heard of like things like, oh, the nutrients in a salmon were transferred to a bear, a grizzly bear that ate it, and then they found those nutrients in like grapes in a vineyard. It's sort of the same concept. These amphibians are really important to that sort of nutrient cycling. So my objective here was really just to see, I'd already had a previous paper that established that yes, these wood frog tadpoles, Lithobates sylvaticus or Rana sylvatica, take your pick as far as the name, but that they're having important roles as predators of freshwater invertebrates. Here, though, I wanted to sort of expand on that and say, okay, but does body size of the tadpole, does habitat shading, does that influence how much they're engaging in predation, how much they're eating? So here for my field collections, again, I did do this in Kentucky, but we are a border state, so I feel like it's pretty close. So, so we had in the Daniel Boone National Forest, which you can find, central Kentucky, on the left there, we can see, and the right, I visited 10 ephemeral ponds. If you're not familiar with that term, it's just temporary ponds, these ponds that become very waterlogged in the spring when things thaw and it rains. Like these temporary water bodies can form like the photo on the right. Actually, this pond on the right, pond, quote unquote, had not been discovered before. So I named it Brady's Pond, which I think is pretty cool. But I did that. So, but then on the left, you can see if you zoom in, I think it was at a later date, but it was in this pond. If you zoom in, you can see on the left there, that is just one, big wood frog egg mass. There's different specific sort of like submasses, I guess you could say, in there. But this was all one, or clutches, I guess you could say. But this was all one big mass, and my boot is in there for a size reference. But these females can easily lay, 1,200 eggs, just one female in a sitting. So this could have all been, less than a dozen females produce just that many eggs. And they can do this, just as a quick aside, because they lay the eggs and they're pretty small, but they take up water so that they don't take up much space in the frog, but once they lay them in the water, they balloon up and then they get really, really big like that. As far as that jelly that's surrounding the black eggs. But what I did was I collected 1 liter, just a 1 liter bottle's worth of these wood frog eggs from 10 ephemeral wetlands in the Daniel Boone. So I had 1,000, we had about 100 eggs per bottle. So we had about 1,000 eggs total that we raised. And then as far as the experiment here, what we did was we wanted to establish a couple groups here. So one, we wanted to establish, how does body size affect things? So to establish that, a lot of folks do differences in density to establish different size groups. If you do a high density of tadpoles or eggs to begin with, then it's going to be high competition and each individual is probably going to be pretty small. But that's not what we did here. What we opted for was to do temperature mediated size differences. So on the left there, you can see that we took half the tadpoles. We put them in these little, they were literally deli trays, and we put them in a fridge, essentially, a growth chamber at a lower temperature than the field. So these ones that were in the small group, they ended up being smaller. They were colder, kept at lower temperatures, while the ones outside were warmer. They hatched first, they got to take advantage of food first, and they developed larger than these ones. Then we put the cold ones in, and they were smaller throughout the experiment. But we also wanted to establish on the right there different shading groups. So we had 100 of these little, if you see those black containers there, we had 100 of those total. 80 of those we put tadpoles in. The other 20 we did not, just to control for, what's the effect of tadpoles. And for half of all those tanks, so for 50 of those 100 tanks, you can see on the right there, we shaded them with 88% shade cloth, just to imitate a sort of pretty natural canopy coverage. So we had half all the tadpoles were small, the other half were large, and then half in each, half of each of those size groups, half are shaded, half are not shaded. And the last thing I'll point out here is that you do see in each of those black little 20-gallon mesocosms, there is what looks like if you ever worked in fast food, looks like a fryer basket, sort of like that. And we kept some of the tadpoles inside just to see, they can't interact with any of the invertebrates outside. So what's their non-consumptive effect? And then the other half of the tadpoles we kept outside the cages, so they could actually interact with inverts, and so they can have consumptive and non-consumptive effects. So here's just a layout, pretty much what I just said. We had each of these circles just represents one of those mesocosms, one of those 20-gallon tanks. In the 20 on the left there that say control, there were no tadpoles. Half of those were shaded, half of those were not shaded. And then in the squares on the right, you can see that those ones actually had tadpoles. Each tank had 10 tadpoles to begin with. Some died, we couldn't help that. But some are large, some are not shaded. And so we have all those groups established. And we also, you can see in gray, where it says consumptive plus non-consumptive, those tadpoles were free swimming. So they could both eat inverts and they could have non-consumptive effects, like producing, it's kind of like the equivalent of a smell like that we would have, like the inverts can smell a cue that tadpoles are producing. Whereas in the black circles there, those are just the non-consumptive tanks. So in those ones, tadpoles were in cages. They couldn't eat invertebrates, but they could still produce cues and have an effect in that way. So for statistical analyses here, I'll pretty much just, I'm not going to go in the weeds with this, but I'm just going to say, we looked at pretty much how the presence of tadpoles, whether they were caged or uncaged, as well as body size and habitat shading. How was that affecting the abundance of invertebrates in those tanks? And we broke that up into the big inverts, we can call them the macroinvertebrates, and the zooplankton, the tiny stuff that really is not swimming very well. They're just sort of there. But we broke that up into those two sort of size groups into taxonomic distinction. And we also took a look at, is things like habitat shading, how's that affecting environmental variables? We can say, biofilm mass, dissolved oxygen, relative fluorescence, temperature. And lastly, we just looked at, how are these treatments influencing, especially habitat shading, how's that influencing the growth of these tadpoles as well. So for results here, this is I'm using an updated figure here because we've currently had 2 rounds of revisions on this paper, and the reviewer has requested that we add these letters and have all these different bars, and so there's a ton of different stuff here. But really just what I want to point out is we have the treatments denoted on the x-axis at the bottom there. We have your average abundance of macroinvertebrates per mesocosm on the y-axis on the left there. And then the dark gray bars represent tanks or groups that were shaded, and the light gray bars are those that were not shaded. Really all I want to draw your attention to here is that if we look at that tallest bar there, so those are the number of macroinvertebrates, those big invertebrates on average that you would find per tank for caged tadpoles that were large and did not have any shade. So there you can see it's really high. And you can then see, if you turn to the uncaged groups there, we can see if we look at the small and large dark gray bars, those were small and large uncaged tadpoles that had shade. So we can see there's a significant difference there, and that maybe that significant difference is because for that tall bar there, those are no shade. There's no shade to be found, and those tadpoles, despite being large, could not directly interact. They were caged. They couldn't actually get at the invertebrates. So, but in the dark gray bars on the right there and uncaged, those tadpoles could get to invertebrates, and those tanks were shaded. So it very well could be that these tadpoles, if there's low light, if it's shaded, there's not much primary productivity. There's not much algae or biofilms to feed on. And because of that, they really have to hammer these invertebrates. So that helps to explain why, even despite there being large tadpoles in that caged, unshaded group, that tallest bar, that we see much higher abundance of macroinvertebrates there compared to those shaded groups there. Here is just the results of an anosim that we ran. And we can see here on this ordination plot, really what I just want you to take away from this is that if you see in black there, these are names of different invertebrate groups, like Culicidae there in the center. Those are mosquitoes. Chironomidae, those are the non-biting midges. And I'd be happy to answer the questions about those. But what we can see here, though, is that if you see the independent variables in blue near those invertebrate groups, that means they're closely associated with them. So here we can see tadpole presence control. That means tadpoles were not there. It was a control, so tadpoles are absent. We can see that that's pretty closely related to Trichoptera, worm eggs, Chironomidae, Culicidae. So that what that pretty much is saying that you tend to find those groups when tadpoles aren't there. So they're probably tadpoles are eating those groups, and we have evidence to suggest that. Whereas in the other vein, if you look, say, in shaded, so there's only one group up there on the top right that if it's shaded, you tend to find them more, the nematodes. That was pretty rare. But really just showing that you tend to find most of these groups when there's not tadpoles, and especially when they're not uncaged and they're not large, you tend to find more of these. And then this figure really is just for zooplankton. It's the same as the previous figure, is sort of the layout. And really all I just wanted to show is if you look at again, that tallest bar where it says A, we see that those are caged small tadpoles that had no shade. And you see more zooplankton there versus on the right there when tadpoles are uncaged. And either there's shade or there was no effect when we had small tadpoles that had no shade. But you can see there's more there than when we had uncaged small tadpoles shade or uncaged large tadpoles shade or no shade. So really just what I think a takeaway from this is just that, again, for zooplankton too, if we have tadpoles that can actually, there's not much in the realm of non-consumptive effects here, but if the tadpoles can actually get at the zooplankton, and especially if it's shaded, and it's probably forcing tadpoles to have to eat more animal matter because there's not much light supporting plant growth, algae growth, then we're probably going to see these big differences because the tadpoles are going to have to eat more of these inverts. And lastly, there's just I just have a few I think simpler slides here because these, in all honesty, this is what the reviewer for our paper ultimately requested, and we made those, and I myself have a bit of difficulty interpreting that at times. But I think here on these figures, this is just showing that on the x-axis we have tadpole size class, so small and large. And then on the y-axis on the left there, proportion of tadpoles that surviving per mesocosm. And again, dark gray shaded, light gray unshaded. We can just see here that yes, our size treatments were effective, and that tadpoles that we put in that were larger stayed larger at the end of the experiment. And we also see that there really was no effect of shading on tadpole survival here. So you see, like within a size group, we didn't see any significant difference in survival based on whether the tanks were shaded or not, because the letters are matching there. But if we look at just growth, or say as a metric of growth, final tadpole body length, we can see here, again, we have tadpole size class on the bottom x-axis there. And then we have the final body length on the y-axis. We can just see, I can just sum this up for you and just say that, again, we see that these animals that were, actually, let me rectify something real quick. Larger, this was showing that larger animals survived better. Here is actually what's showing that these larger animals stayed larger till the end of the experiment than smaller animals, but also showing that when it was unshaded, you do see a larger body size, a greater final tadpole body length than when it was shaded. And that could be due to a variety of factors. Could be due to vitamin D, needing vitamin D from sunlight, or rather sunlight to effectively process vitamin D, synthesize vitamin D. That influences growth. Could be that you need that additional sunlight to have enough food to actually grow and reach those body lengths. Could be a variety of things. But these animals did reach a larger size when there was no shade, when it was lighter. And this is effectively showing the same thing, just for the only difference here is that it's final head width. So whether it was body length or head width, if the tadpoles were initially larger, they ended up larger. And if there was more light, they ended up larger. So then just closing here. So how do body size and habitat shading shape consumptive effects on invertebrates? So what we generally saw was that, and those were looking at pairwise differences in those sort of more complex figures. But what we effectively saw was that macroinvertebrates and zooplankton, you generally saw fewer of them when you had less light and when you had these larger tadpole predators. And some possible explanations for this are just that if you have a larger tadpole, just intuitively it makes sense they can eat more, but also if any prey has sort of a defense mechanism, particularly an inducible defense mechanism, that is just to say maybe it doesn't have that defense all the time, but when it detects a predator cue, it may trigger it to develop that defense mechanism, such as in Daphnia, a type of zooplankton, or colocerines, I should say. They can develop these defensive head spines if they detect fish cues or tadpole cues. If you're a larger predator, if you're larger tadpoles, then those defenses really don't, make that as much of a difference. If you're smaller and you're biting into something that has these tiny spikes all over it, yeah, it's probably going to dissuade you from eating these things. But if you're predator's just really large, then those little spikes aren't going to make as much of a difference. And then in the sense of more shade, if we have more shade, we have less light, we probably have less available algae and other sorts of biofilms for these tadpoles and invertebrates to feed on. So that's probably going to force tadpoles to feed on more animal matter than those algae and biofilms. So effectively here, just saying that bigger tadpoles are going to be eating more animal matter. And if you have less light, we're going to be seeing that there's less algae. And again, your tadpoles are probably going to be having to eat more animal matter. And then for non-consumptive effects, we didn't see much in the realm of non-consumptive effects here, but we did see an overall stronger non-consumptive effect in terms of just predators, the tadpoles here being there, and maybe their cues having an effect on invertebrates. That was stronger on zooplankton, the smaller guys, than the macroinvertebrates. But in either case, I mean, they could have been mediated in several ways. For one, larger predators likely produce greater concentrations of these chemical cues. If you're, regardless of what animal you are, an aquatic animal, if you're bigger, you're going to be probably producing more of these cues, per square inch of your body, like through your skin. And that may have greater impacts on just the abundance, the foraging, the reproductive rates of various prey that are detecting these cues. But also, it could be that invertebrate prey are altering their foraging patterns in the presence of predators if visual acuity of either the predators or prey is influenced by habitat shading. That is just to say that, if it's darker, then both predators and prey are going to have a harder time seeing each other. And that could pan out in a variety of ways, depending on how much each group relies on light to see things or to detect things in general. And my final sort of content slide here is just showing that, how does habitat shading mediate predatory roles indirectly via effects on body size? It could very well be that tadpoles, they, in our case, they grew faster and larger in unshaded mesocotyledons when they had more light. And again, that could be taking place in a variety of ways, whether it's direct effects of light on growth or effects of light on food that the tadpoles need to grow. Shading may also influence predatory effects on prey indirectly by affecting the growth of tadpoles and the gape size, that is to say, the mouth size, then being the direct determinant of predation. If you have a larger mouth and that's influenced by light, say if you have more light, you develop larger, that includes a larger mouth, then you can eat more. It could be a variety of ways that light is influencing things here. And finally, for roles of tadpoles and invertebrates as predators versus prey are also mediated by size and shading, because here we focused on tadpoles as the predators here. We excluded the sort of larger predatory invertebrates here, like dragonfly, nymphs, and things like that. The giant water bugs, the bellostomatids. We excluded those here. But it very well, in a natural condition, could have been, the tables could have been turned here, in that larger tadpoles are less susceptible to predation and more likely to be predators. If you're larger as a tadpole, you're more likely to be a predator. But if you're smaller than, say, a dragonfly nymph, you're more likely to be the prey. So it could very much go in either direction. Same with mosquito larvae, for example. There's been studies showing if you're a tadpole and you're bigger than a mosquito larvae, you're going to eat the mosquito larvae. But if the mosquito larvae is larger, you're actually going to get eaten. And lastly, tadpoles need proper light conditions for proper development and to be effective predators. So really just saying that, one, size is important, and two, I think we're really highlighting that lighting is important for these animals. And that's just what it says here. Body size and habitat shading are important to predatory effects of these tadpoles on freshwater invertebrates. And in the future, we're just interested in looking into how changes in other factors linked to climate change, such as precipitation, influence the effects of freshwater predators on their prey. And that is just one of our tadpoles there. And now I'll take any questions if you have them. I'll stop sharing my screen.
So Brady, the tadpoles that were non-consumptive, they were basically eating the phytoplankton, is that correct?
Yes, they were. If we had them caged, they were just feeding on biofilms.
Brady, has anyone, I don't know if it's even possible, but have people looked at gut contents of tadpoles to see what they're eating?
I have. I did that in that first paper. That was the first like half of my master's thesis, and then this has been the second half, what I just talked about. And I did do that for the first half, and you do see like they are really hammering these zooplankton populations, like ton of cladocerans, ton of copepods are in there. And to a lesser extent, but you still see them, things like midge larvae and a oligochaete, like a freshwater earthworm relative. You see those in there too.
It's nice to have definitive proof in addition
Yeah,
to the second
it,
step.
it's just that a lot of folks, like I think that's definitely a key piece of evidence. A lot of folks will say, well, it's just a snapshot and you don't know at one time period, you don't know what they were eating at other times and how much. So it's really that's what makes things complex here.
I was going to ask too, one of your variables was fluorescence. Is that basically like the measurement of visibility, how much light there is to see, or is that something else?
That was essentially a measure of primary productivity, in that we took a handheld, a like portable probe out there, and effectively just took those relative fluorescence measures to as a proxy for okay, how much green algae is in the water, and it's just sort of a quick and because we weren't incredibly interested in rates of photosynthesis, but relative fluorescence gives you a sort of quick and dirty metric of how much photosynthesizing is going on by that green algae.
It's you know, I was fascinated with the concept, and in the past, I was involved in a number of wetland mitigation projects and restoration projects. What is the future of wetland restoration and recreation in the United States is a little bit cloudy right now. The Clean Water Act was interpreted over a series of legislative and legal challenges, to a point where in just a few years ago there was extensive remediation taking place in association with any impacts on wetlands. And ultimately, the definition of a wetland was and the effectiveness or the authority of the Clean Water Act has been changed significantly to now isolated wetlands are no longer protected. Okay, and that's just a precursor. What I found is interesting is during the heyday of wetland remediation, if a forested wetland was adversely impacted, cleared and filled, there was a process where a company or the organization that was filling the wetland could go and create or contribute to wetland remediation banks. All right. And there was a lot of work done to try to classify and grade wetlands based off of their plant communities and soil types and whatnot. There was no significant research available at that time to try to quantify the impacts on invertebrate communities. And so there was a lot of speculation about that, associated with fishless habitats. That was one of the big buzzwords for many years. And I think that this work and comparable work is really key in illustrating there's a lot of subtlety involved, especially in ephemeral wetlands. that if we're true to understanding, our efforts are truly intended to understanding the significance of these very specific habitats, there's a lot of work that needs to be done to get a handle on what all is actually occurring in these site-specific wetland types.
Yeah, I think as far as specific wetland types, I think that the types of these ephemeral wetlands, these temporary wetlands that these animals are occupying, and granted, most of them for a limited period of time, they're pretty much just larval stages of a lot of things, but they are still really important to environments in which you do not have a lot of permanent water bodies, as far as cycling of, say, leaves are breaking down in those ephemeral pools, and then the larval organisms, whether they're amphibians or they're mosquito larvae or whatever, are then taking those nutrients, because they're in large part, a lot of, at least part of their diet are bottom feeders, suspension feeders. They're taking that and out into the environment, and if without these wetlands that a lot of people I think are ignoring, you wouldn't be getting that nutrient transfer, because if you just don't have these big lakes and ponds that we're typically trying to save, then these temporary pools are what you have. So they still, I think, play a valuable role.
Well, I think it's huge, and unfortunately, as the mitigation business developed, it was much easier to do large mitigation projects as opposed to small ephemeral pool recreation or creation. And so you would see these isolated wetlands being replaced by large aggregated projects. So, instead of quarter-acre ephemeral wetland, swamp forest kind of thing, you would see them all being replaced with a large marsh complex, completely different functions. And I think your work and other work like this illustrates there's much more subtlety involved, and we got a ways to go.
It is, and I'm glad you brought that up, because they're, as far as like mitigation projects or companies or states at the state level developing habitats to, say, make up for some sort of construction or some sort of habitat alteration. In a lot of cases, you do build habitats that are not what you destroyed. You destroyed, say, 100 acres of forest habitat that had however many of these ephemeral pools that served particular roles. But then to replace it, you built however many ponds, these non-draining ponds that allow, things like newts, wood frogs might breed in them, but then things like newts or whatever can come along, they'll just eat all the tadpole eggs, and it really is not serving the same role.
Yeah, it was very fascinating. When I was working at Lake Metro Parks, we built a number of these forested wetland complexes in mitigation projects. We actually went from being a teaching site for the Army Corps of Engineers to a demonstration teaching site to that same project no longer being permittable within a 6-month period of time because of the political involvement of wetland mitigation banks. And it was very interesting to see that kind of stuff unfold. And we had nothing to fall back on, like work that you have done to try to argue that. And so it was going to these larger mitigation projects. But yeah, fascinating research, Brady.
Thank you. It has been a little bit of time since I've I've gone over this, but I think it's good to get back and review some old stuff. So thank you for the opportunity.
Yeah, you might consider making a submission to the Ohio Biological Survey for a presentation at their conference, which is in January. Our next month's presenter is Brad Lepper from the Ohio Historical Connection. And he's going to be talking about the significance of the Mound City Group being designated a World Heritage Site. And so that should be a fascinating topic and presentation. And Rich, did you have something else you wanted to add there? You're muted, bud. Yeah.
That was great. I enjoyed it. And, you know, once we get the recording, I'll post that very quickly.
And do we have, just to make sure, do we have access to that? I think it might just come up on either Darren or I's. Once we end the meeting, it'll come up and then we can share it.
In about
Yeah, I think one of us should get an email about it.
Okay. Gotcha.
yeah, and
I'll keep an eye out for it in case it comes to me.
it
Okay.
should go to Darren if he was the actual registered host. That email, you can forward that link to Rich, and Rich will take it from there.
All right. Sounds good. Thank you, Rich.
Oh, this was good. I think it'll add to our library.
Yeah, thanks for stepping up, Brady.
Yeah, no problem. It was I didn't really have an opportunity. It's my field season right now. I didn't have a big opportunity to adapt anything from this because a lot of this is coming from my thesis defense. But yeah, again, I appreciate the opportunity.
And I think next, one of the elements that we want to explore on some kind of email chain or in-person discussion is, now that class is getting ready to resume, we're back in session, how do we communicate with the students here at Case Western Reserve and other students about upcoming topics, and how do we invite them to participate? And so I will follow up and send a little prompt out to everybody, and we can put our heads together and try to make sure that we welcome other interested parties into the discussion.
Very good. And next month I'll be on the road up in Maine, but I'll be able to, once I get the email of the recorded presentation, I'll certainly be able to post that, you know, right away from there.
All right, thanks everybody.
All right, sounds good. See you all next month.
Thanks. Have a good rest of the day.